Is osteoarthritis simply a wear and tear of the joints? Well, only partly. Osteoarthritis is a complex degenerative joint disease causing the protective cartilage on the bone ends to lose its function. While risk factors do include overuse, aging, and direct injuries, they also include inactivity, metabolic dysfunction, and systemic inflammation. It turns out that it is a widely held misconception that activity necessarily worsens osteoarthritis, and lower activity levels might even counterintuitively compound symptoms. Osteoarthritis currently affects more than 528 million people worldwide, an increase of about 113% since 1990. Due to aging populations and increases in obesity and injury rates, this is only expected to continue to rise in the future. And evolutionary medicine might hold the key to understanding osteoarthritis and possibly improvements in treating it. In this video, I'll be examining 3 main evolutionary hypotheses explaining the prevalence of osteoarthritis today. Number 1, osteoarthritis is a disease of evolutionary-modern mismatch. Number 2, osteoarthritis is the result of antagonistic pleiotropy, which I will explain later. And number 3, many cases of osteoarthritis can be attributable to a genetic mutation representing an evolutionary trade-off that occurred when humans migrated north towards colder environments. The first mismatch between humans and our ancestors is the fact that humans are bipedal, a trait that allowed us to free up our hands and maximize the use of our brains, but still could have come with some downsides. Quadrupeds can disperse their weight and thus gravitational forces between 4 legs, whereas our knees and hips, the 2 most common sites of osteoarthritis, must bear our whole body weight. There's also much more direct weight bearing done by our spines, which are also prone to osteoarthritis. This is in contrast to rheumatoid arthritis, which is a systematic autoimmune disease that first symmetrically affects the smaller joints of the hands and the wrist, joints that are not weight-bearing. These gravitational forces that contribute to osteoarthritis are even greater during walking, when our joints experiences forces more than twice our body weight, and running, when our joints experience forces that are more than 4 times our body weight. This might have put excess loads on our bones, resulting in significant morphological changes, such as expanded condyles of the distal femur, and other adaptations that likely occurred over 1.6 million years ago. Despite these adaptations, the physical constraints might still cause osteoarthritis later on. The second mismatch is how we use our bodies versus how our Pleistocene hunter-gatherer ancestors used their bodies in the environment of evolutionary adaptiveness, or the EEA. Our current lifestyles are very different from our ancestors in many ways. Not only are we mostly sedentary, but oftentimes when we work out, we can go to extremes that our bodies are not used to. This is reflected in the data. Hip and knee osteoarthritis prevalence is 10.2% in sedentary individuals and greater at 13.3% in competitive runners, according to a meta-analysis of over 100,000 individuals. Interestingly, the group that fared the best was recreational runners, who had an osteoarthritis rate of only 3.5%. This might reflect the dangers of extremes beyond the levels at which our bodies were adapted. Further, it could reflect how our ancestors would have slowly built up resistance and flexibility over time by squatting constantly and remaining active. Our ancestors didn't sit or stand as much as we do today, and research has shown that these squatting postures activate the leg muscles to a greater degree, which might have been protective for osteoarthritis. For sedentary individuals, lower quadriceps muscle strength is associated with a greater risk for incident symptomatic knee osteoarthritis. Further, this can cause greater problems such as obesity and metabolic dysfunction. This compounds osteoarthritis in 3 ways. First, it puts extra weight and force on the joints. Second, it generally leads to greater inactivity. And third, it increases cytokines and general inflammation, which is further damaging to the joints. Clinical data have shown that a 10% reduction in body weight in obese individuals resulted in moderate to large clinical improvements in osteoarthritis joint pain. It has also been shown that total inactivity can be particularly harmful in both humans and rat studies. Patients with spinal cord injuries show markedly rapid loss of articular cartilage, about 20% loss in 1 year. Similar joint cartilage deterioration was observed in rat knee joint immobilization studies as well. Meanwhile, for highly active individuals, it is likely the case that very repetitive motions in endurance sports or quick turning motions that can often result in tears of the ACL in other sports can cause osteoarthritis. These ACL tears or accompanying meniscus tears were likely uncommon in the EEA or the environment of evolutionary adaptedness. While it is generally known that long-term overuse can result in worsening osteoarthritis, it is not often appreciated the role of these kinds of acute orthopedic injuries in osteoarthritis development. For instance, an ACL tear can cause the collagen to loosen, which allows proteoglycans in the joint to attract water and swell. This leads to subchondral bone thickening, more pressure on the joints, osteophytes, also known as bone spurs, greater synovial inflammation, joint swelling, pain, and instability. Overall, it is thought that an ACL tear ages the joint by 30 years, and the incidence of post-traumatic osteoarthritis has been assessed at a staggering 87%. Basically, 9 out of 10 patients with an ACL tear will get osteoarthritis. So, while high mechanical stress might be an approximate explanation for osteoarthritis, changes in our activity patterns might be a better ultimate explanation. Further, ACL tears may be part of the reason for the increased risk of osteoarthritis in women, since women are also more susceptible to ACL tears. Osteoarthritis seriously affects an estimated 10% of men over 60 years old, by 18% of women over 60 years old. This is a stark difference that begs an evolutionary explanation. Part of this might be explained by the difference in ACL tears between female and male athletes. Female athletes are up to 8 times more likely to tear their ACL. It is argued that this is due to several factors, including wider pelvises, increased quadriceps angle, commonly called the Q angle, steeper tibial slopes, and hormonal changes such as estrogen or progesterone, which are correlated with greater joint laxity or instability. Overall, these differences contribute to poorer joint congruence that promotes cartilage wear. In this way, osteoarthritis can be seen as one of the evolutionary trade-offs of bigger brains, which require wider pelvises for the birth of babies with comparatively larger skulls. Altogether, this data suggests a significant mismatch between how women evolved and the modern environment with its novel stressors, such as sports, or at least some of the evolutionary reasons behind osteoarthritis prevalence. Next, some suggest that osteoarthritis is the result of antagonistic pleiotropy. Antagonistic pleiotropy refers to the evolutionary theory wherein a gene may confer beneficial effects on fitness, like increased reproduction, early in life, but causes detrimental effects, like disease or degeneration, later on. This results in a selection shadow, whereby natural selection is strong before reproduction, but weak afterward. This explanation is generally used to explain the evolutionary mechanisms behind cellular aging or senescence and the many diseases associated with old age. For example, Alzheimer's disease, Huntington's disease, cancer, coronary heart disease, and so on. There are several pieces of evidence to suggest this is how osteoarthritis behaves. First, osteoarthritis is greatly associated with chronological age, how long one has lived. However, it is even more closely associated with physiological or biological age. A 2024 cross-sectional study with over 30,000 participants demonstrated that an increase in clinical parameters of biological aging based on blood tests was associated with a higher risk of osteoarthritis. This tells us that it is likely a biological process rather than a simple wear and tear physics problem. This is bolstered by the fact that osteoarthritis consistently onsets at around the mark of the last third of an animal's lifespan, and this has been shown in several wildly different species, including mice, cats, dogs, and humans. This suggests that while it coincides with the natural processes that cause aging, supporters of the osteoarthritis antagonistic pleiotropy hypothesis contend that osteoarthritis results from a dysfunction of chondrocytes, the specialized cells found in cartilage that maintain the extracellular matrix in low oxygen avascular environments, like the articular cartilage at the distal end of the femur in the knee. They argue that the genetic program that turns these chondrocyte cells on and causes them to hypertrophy or grow in size are typically beneficial in normal early development. In normal early development, this program allows the long bones to grow normally through endochondral ossification, which is when cartilage is replaced by bone. However, the same genetic program can be pathological if reactivated later in life and can cause osteoarthritis, since hypertrophic chondrocytes can result in inappropriate bone remodeling and the development of osteophytes and cartilage breakdown. One article called this an endochondral ossification quasi-program. One way in which this system might be maintained is through TGF-beta signaling. Using bovine, cow, articular cartilage extracted post-mortem, it was shown that this signal disappears without loading, that is, without a compressive force, and is induced by loading with a compressive force. The presence of this signal then maintains normal chondrocyte activity, whereas its loss results in hypertrophic chondrocytes and thus osteoarthritis. It would appear that this may explain how inactivity or immobilization results in greater osteoarthritis, as we discussed earlier. Further, it suggests a possible therapeutic target. Perhaps TGF-beta expression may be increased in order to block pro-hypertrophy quasi-program in chondrocytes in order to prevent osteoarthritis. The osteoarthritis antagonistic pleiotropy argument posits that early chondrocyte hypertrophy is beneficial in early development, but late chondrocyte hypertrophy in old age can cause deleterious effects in osteoarthritis. It is argued that various genes underlie this action, and that some genes that might increase the likelihood of stronger, more functionally sound bones in early life might also allow for osteoarthritis later on. This theory seems plausible, considering that between 39% and 65% of our osteoarthritis risk is explained by genetics based on classic twin studies. It is especially plausible considering that osteoarthritis risks rise sharply after menopause, when women are no longer fertile, and this is likely mediated by declining estrogen levels. Postmenopausal women are nearly twice as likely to develop osteoarthritis than men of the same age. Further supporting this is the idea that perhaps older members of a tribe with osteoarthritis, although rare in the EEA, might still have been able to contribute to their fitness of their offspring or their grand offspring through helping with child rearing or aiding in passing down traditions and valuable knowledge. This might be an example of kin selection or the good grandmother hypothesis. In this way, genes contributing to osteoarthritis incidence might have continued to persist in the gene pool, if not fully selected against. Looking more closely at the genetic basis for osteoarthritis revealed a third hypothesis, an evolutionary trade-off resulting in a gene, now common in many populations, that increases the likelihood for osteoarthritis. Specifically, this trade-off involves selection on the gene GDF5, which plays a key role in skeletal growth and joint formation. A 2017 study looked directly at the effects of functional variants associated with GDF5 in mouse development, and also tied this with GWAS, genome-wide association study analysis, to identify specific gene variants and haplotypes. This research identified a regulatory enhancer near GDF5 called GRO1 and showed that a single nucleotide variant reduces the activity of the gene in the growth plates of developing long bones, producing shorter bones and reduced stature. Homozygosity reduces height by approximately 1 centimeter. Genetic and population analyses demonstrate that this variant likely rose to high frequency through positive selection during human evolution, particularly in Eurasian populations and Neanderthals. Research shows that the derived allele frequency is about 60% in Europeans, about 70 to 80% in East Asians, and only about 40 to 50% in Africans, hinting that it could have something to do with increases in latitude. The likely adaptive advantage was related to cold climate survival during migrations out of Africa roughly 50,000 to 100,000 years ago. Individuals with shorter limbs and more compact bodies would have lost less heat and been less at risk for frostbite. They may have also been less susceptible to fractures or falls in icy environments, making this skeletal configuration advantageous in northern climates. However, the same regulatory variant is genetically linked to alleles that increase the risk of knee and osteoarthritis, raising osteoarthritis risk by roughly 1.2 to 1.8 fold in modern populations. Hence, the allele that improves survival and locomotor safety in cold environments also alter joint morphology and cartilage biology in ways that predispose individuals to degenerative joint disease later on in life. This conforms with data from the Rotterdam study, a prospective cohort study that has been ongoing since 1990 in the city of Rotterdam in the Netherlands, with over 15,000 subjects aged 45 years old or older. One 2008 article showed that one specific polymorphism near the GDF5 gene seemed to be the ancestral trait and not the derived trait. it seemed to have the opposite effects as GROW1. For homozygote women, it resulted in a 37% reduction in hand osteoarthritis, 28% reduction in knee osteoarthritis, 1.1 centimeter taller height, a larger hip axis length, and a 29% increased risk of incident non-vertebral fractures. Thus, the cold trade-off hypothesis seems plausible, especially considering how, without the derived shorter phenotype, a person is more likely to have a fracture, something that could have been deadly in the EEA and greatly reduce fitness. However, it does not necessarily explain height differences between different groups. For example, according to available 2016 CDC data, height differences between Caucasians and African Americans in the United States is very small, with non-Hispanic white men being 1.9 centimeters taller than non-Hispanic black men, and white women being 0.7 centimeters taller than black women, the opposite of what we would expect based on the GDF5 allele rates. However, this could reflect sexual selection for taller mates, external developmental factors affecting height, or a number of different factors. Further, there is very little data on GDF5 enhancer mutations in Alaskan Native populations, but one study showed the age-sex adjusted prevalence of self-reported arthritis was high in Alaska and low in the southwest U.S. As compared with the U.S. population, there was a 26.1% chance of reported self-reported arthritis in Alaskan populations and a 16.5% prevalence of osteoarthritis in the southwest U.S., as compared with the general population of 21.5%. This could hint at either a similar GDF5 enhancer mutation, as described in the 2017 study, a completely different genetic mutation that was selected for under similar cold northern constraints, or totally different reasons. This necessitates further research. Further complicating this paradigm is the contention that weather changes and cold temperatures can make osteoarthritis worse, although some doubt these claims. It's unclear if or how these factors are implicated in the GDF5 gene trade-off hypothesis. In conclusion, the evidence suggests that osteoarthritis is not merely a mechanical failure of aging joints, but an evolutionary byproduct of how the human body was built, how it ages, and how it now lives. The mismatch hypothesis explains why osteoarthritis may be especially common in modern populations characterized by inactivity, obesity, metabolic dysfunction, and novel injury patterns. The antagonistic pleiotropy hypothesis, however, offers the strongest general evolutionary explanation because it accounts for why selection would tolerate traits and pathways that are beneficial early in life but harmful in old age, such as the later life reactivation of chondrocyte hypertrophy programs. The GF5 trade-off hypothesis adds an important layer of specificity by showing how a particular adaptation that may have improved cold weather survival and skeletal robustness in the past could also increase osteoarthritis risk in the present. Importantly, these explanations are fully compatible rather than competing. Genetic trade-offs such as GDF5 can be viewed as one mechanism within antagonistic pleiotropy, especially considering that GDF5 is part of the TGF-beta superfamily, the molecule that inhibits chondrocyte hypertrophy. Further, modern environments may intensify the late-life costs of these inherited traits while minimizing the importance of previously advantageous traits that helped us to withstand exposure to the elements in the EEA. Thus, osteoarthritis is best understood as the combined result of evolutionary trade-offs, selective shadow, and modern mismatch. Still, if one framework best explains why osteoarthritis persists so widely, antagonistic pleiotropy is likely the strongest because it captures the central evolutionary reality that natural selection is weakest precisely when osteoarthritis tends to emerge. It's my hope that future research continues to produce and evaluate evolutionary hypotheses, not only for osteoarthritis, but for many other diseases in order to shed light on them and contribute to cures. Well, that's it for today's video. Thanks for sticking around till the end. Make sure to subscribe, share the video with anyone who's interested, turn on that notification bell if you want to be notified whenever I put out new content, and comment what questions I should look into next. See you next time.